Welcome back to the lecture series in cell culture technologies. So, today we are into the fourth lecture of the second week. Last lecture, if you recollect, we talked about or started talking about the design or the layout of the cell culture facility, and there are certain points which you have to keep in mind. So, today what we will do, we will just first of all let us put that layout in front of us and keep on adding the components which we will be needing. Because we are into week 2, lecture 4. Okay. Design and layout of cell culture facility. And in the last class I told you, let us assume that this is our working area, something like this. Suppose this is the entry can change, it could be in the corner. So, or if you could make an entry in the corner, say for example, you have such option, you can make in the corner or somewhere out here on this side and this side, then you have few advantages. Then your wind flow or the current, air current will remain contained in one side, okay. But nevertheless, that is not in your choice, you will get a room and you have to really set up the facility or couple of rooms whatsoever, okay. You are not really doing the imaginary work to construct a room. So, what he talked about is that either in, so let us A, B, C, D, the 4 corners of a room, and we talked about either this corner, okay, where you will have either I1 or this corner I2, okay, where you will be placing the incubators possibly, and you will have a countertop something like this. That way you will have sufficient surface area, okay, something like this, and adjacent to it you have to keep a gas cylinder. So, you have location for the gas cylinders, and obviously if you are keeping the incubator like this, so you will have a little bit more of working bench like here. So, the door will possibly either will open in this direction of this incubator or in this direction depending on. So, you have to ensure that how much area of this side tables you are going to make, and if you are doing it in India, so ensure that the height of these countertops what you are developing should match with the average Indian height, which is around something between around 80 centimeter or something. I mean, that is where you have to. So, you just look through it, because sometimes we make those heights pretty tall and it has its own set of issues. Okay. So, now I have already talked to you like these are the places for the incubator, and we talked about that just outside the door or somewhere out here you will have the place for the extra cylinders, which should be tied up or kept in proper frames so that it does not fall down, and you needed to have a cylinder and cylinder trolley to carry the cylinders inside and outside, and prefer to follow something like this. If you take the cylinder, you have to take the cylinder to this spot, follow one track, do not kind of mess up the center of the lab, the whole surface area what you are having. Then I told you that you will be having a nitrogen devar, you should have 2 nitrogen devars, one to store the cell lines, the other one as to bring the liquid nitrogen, because you cannot carry this parent stuff every time. So, you have to have a space, because every day you have to replenish nitrogen. So, you have to figure out where you want to put the nitrogen storage. Okay. Next important thing is that you have to have a sink along with a shower. This is a call you have to make. So, you probably want to have a sink somewhere out here before you enter to the main frame, which is you wanted to have a sink and a shower. Okay. Now, here at the entry port, you could have two sets of doors, one here, one here. The reason for two sets of doors, why I am telling you, is this is say door 1, this is the door 2. So, you come here, you leave your, so you have to have a shoe rack here, where you will be leaving your shoes, and you have to put on the lab coat or the lab gown, whatsoever you are following. So, you have to have a hanger and everything for the lab coat or the lab gowns. Then you have to have the place for the mask, and especially those people who have hair falls, irrespective of the gender, should put the cap. So, you should have a head cap, and it is kind of a good practice, irrespective of the fact that whether you are having hair fall or not, to put a head cap, and then here to put on the gloves and everything. And you should have a trash here, because most of the time these will be disposable stuff, your gloves and all these things. So, you should have a trash which should be clean every day. Whereas, if you are entering here, so you are kind of dressed up and you are entering through the main door, you prefer to have something called a wind curtain. Which will be a wind curtain, which will be, there will be moving like this from vertically down. Okay. These wind curtains are a very effective tool to ensure that no unnecessary material kind of passes through it to contaminate the system. Now, some people, what they do something very interesting. They kind of split the lab into two parts, like with the main frame like this, okay, at two level. So, this is the level. Say for example, you wanted to have, so you can make it of glass or something, or you can use even these walls for your storage or other purpose, or you can keep a microscope on these tables. Suppose you are splitting the whole facility into two parts. Okay. Say for example, this is the outer culture room and say inner culture room. I will tell you why such demarcation may be very essential. Inner culture room. Okay. Now, see for example, so you have the sink and shower I have already mentioned. So, say for example, like you are entering through this door, you get all dressed up. So, this is in blue, I am showing the dressing area, you get all dressed up. Then the next thing, you enter inside the lab. So, there are, okay, here I just made a small mistake. Do not put on the gloves as you are entering. First of all, keep the gloves in the lab coat. Come to the sink, you wash your hands, and you should have a place for disinfectant here. And once you are done and you wipe your hand, you put on the gloves here. So, you carry the gloves from here and you put on the gloves here. Okay. Now, you are all set to enter the facility, and please ensure personal hygiene. The more I like this is something which you should religiously follow. If you are not well, if you are coughing, it is better, or rather strongly advise that do not get into the facility. If you have infections, do not get into the facility, because you may spoil the work of your colleagues. Okay. Apart from it, the personal hygiene is very important. Just ensure that you have taken necessary care about yourself, because we are the major source, or the workers are the major source of contamination into the system. Okay. So, this is where you have the disinfectant and everything. Say for example, your lab has a primary culture needed. So, where you have to dissect animals. Obviously, dissecting animals essentially means that there will be significant amount of surgery, anesthesia, surgery involved. So, which is at times can be a source of contamination, or many a times it is a source of contamination. So, in order to do that, you would prefer to have the facility kind of split up in such a way that you minimize your contamination issues, or you maintain your cleanliness in a better way. So, let us take an example. Say for example, your lab works on deriving tissues, brain tissues, or cardiac muscle, or skeletal muscle, or liver tissue, or some other tissue from the rat or a mice. Okay. So, the best idea is that, say for example, so all these rats and mice, everything comes in different kind of cages and everything. So, first of all, they arrive from the animal house. So, you have to have a designated animal house, and having said this, let me mention here, you need it for any kind of animal studies, you need ethical clearance. So, ethical clearance essentially means your institute should have a body which essentially have to have a veterinarian who is certified to tell you that what are the ethical practice of sacrificing animal, what are the basic rules and norms and regulation, and which is further stamped and endorsed by specific government dignitaries or government individuals who have been appointed by the local authorities or by your institute or something. So, there is a whole protocol about animal ethics. Okay. So, that ethical clearance is very essential. So, wherever and whichsoever institute you are, look for that committee which takes care of the ethical clearance. So, you have done all those things, I assume, for a minute, and you bring your animal to your facility. Okay. So, for example, so obviously now animal won't go through all the lab coat or everything, you are just bringing it straight. So, this is the catchphrase. So, you are getting the animal, you have to dissect it. So, and knowing this fact, so you have decided that the lab is divided in two parts. So, what you can have is, you enter here, leave this. So, you can have a out here possibly, or in this corner, which may be a better idea, somewhere in this corner where I am shading now, you could have a laminar flow hood, a small laminar flow hood. So, what is laminar flow hood? Laminar flow hood is essentially a rectangular or circular structure, which where the air, so something like this. Let me draw it and that will make more sense. So, say for example, I am drawing a box here. Okay. It has its own dimensions, I am not getting into that, and many a times you will. So, this is the box and this is a, so most all the sides are of glass or mostly synthetic plastic, transparent plastics. So, there are two ways, either you will see an extension on the back of it, or either it could be horizontal or it could be a vertical. So, on the top of it you will see a kind of a enclosed structure. So, the way it works is that there is an air current which comes down like this. Okay. This is force of the air current. So, it ensures that whatever you are doing, you are not contaminating the system, or the air current may flow like this. Either crisscross or upside down. Okay. So, automatically to make the air flow, you will have those fans either rotating from the top or rotating from the side, and there are several permutation combination into it. So, you could have either vertical laminar flow or horizontal laminar flow hood, and all the work flow hood, it could be either vertical or horizontal. So, the vertical ones have a additional attachment at the top, whereas the horizontal ones have on the sides or on the back, okay, mostly on the back. Okay. So, now the way something like this, horizontal ones are working like this, the air is coming from the back, just trying to show you like this. Okay. Whereas, from the top it is coming like this. So, say for example, there is a hood in front of me, either the air will be coming out from the top like this, or the air will be coming out like this, pretty much hitting me. So, that way what will happen, whatever work you are doing, any kind of aerosol or anything will be pulled out, and these rooms are pressurized in such a way, things are being pulled out from them. These are almost like a clean room. So, the whole concept is like a clean room. Okay. So, if it is a vertical one where the air is coming from the top to bottom, the problem is they are slightly taller, and you have to ensure that this vertical one gets inside the facility without any hassle, because at times so happen, with the year through experience I have seen that the vertical laminar flow hood gets stuck up at the door, and you may need it to dismantle certain stuff and bring it in. So, please ensure whatever size of laminar flow hood you are getting, whether it is a vertical or horizontal, it should match into the space. So, the smallest of all these, you can even get them fabricated by your local fabricator, which I have done on several occasions. So, they will be around 2 feet. 2 feet means, so 2 feet like this, your working area, single individual, 2 to 2.5 feet. So, it will be a cube of 2 to 2.5 feet, a cubical structure, something like this. Like this, 2 to 2.5 feet cube, which are the smallest one, where one individual can work without disturbing anybody, but there is no option for the second person to sit in this. What if you are clever, you could have 2 of them side by side, that way 2 people can work absolutely individually without disturbing each other. This is what I am talking about when you have multiple user, or some people have done, I have done this earlier, you have one horizontal one which is something like this, where 2 people can work, but the 2 people in that case, say for example, this is P1, this is P2, 2 people can sit, but the only problem is that, so there is no barrier between the 2, it is on the common table you are working. Okay. So, I will leave it up to your choice how and what kind of laminar flow hood you want. But in nutshell, laminar flow hood could be classified into two categories. The take home message is very clear. One is a vertical, where the air is moving from the top to bottom, and the airflow, it's like exactly like an air curtain. So, air is flowing like this and out, like this and out. Okay, you are sitting outside, right? Or the second thing is that air is flowing from the front, from the back side. What you are looking at the front, from the back side, air is flowing towards you, like this, you know. So, there are modifications, there is something like a semi hybrid and all those kind of things, but do not get into the complexity. Just keep in mind you have two kinds of laminar flow hood, a vertical laminar flow hood and a horizontal laminar flow hood. And having said this, you realize that you will be needing these laminar flow hoods, and just be careful about 2-3 parameters. The size you are looking forward to, size or dimension. One may sound a very trivia point, but it is directly related to your door height. It may, how much soever trivia it may sound, you have to get this laminar flow hood inside the room. So, you have to ensure that they can do it. Just do not rely, because the manufacturer will take care of it. Of course, the manufacturer will take care of it, but there are repairing issues, it may have to be taken out and whatsoever. There are so many problems which comes through it. Okay. Third is number of users. Okay. And frequency of use, how many working hours on a laminar flow hood. So, laminar flow hood will be equipped with 2-3 things. So, by the way, the front, it has a kind of, just like windows you have seen, you can raise the window like this, or you can lift the window like this. They have several such options. You can lift the window, the glass A. So, this will have a UV fixture, they will have a tube light. These are the major fixtures, and they have a switch for the blowers or the fans. Okay. So, try to buy models which are very simple and which are kind of, if you can get them made locally, you are very fortunate. And inside, if you open this part, you will see the filter mesh. They will have the filter mesh, whose job is to trap the impurities or contaminants or the dust and whatsoever. Okay. So, generally before you start working on a laminar flow hood, it is good idea to switch on the UV before that and leave it on for half an hour or so. Much earlier than that, what generally we do is, you step 1, you wipe the surface with alcohol before and after the work. So, it means before you start the work, you wipe it with alcohol properly, thoroughly, okay, 70% to 100% alcohol. After that, the way I used to do, I will share my experience, but I will leave it up to you. I used to switch on the UV and leave it on for 10 to 15 minutes. Followed by that, I get in. First thing you should do, ensure that if you are using, when the UV is on, put on the goggles, otherwise it damages your eyes. So, do not damage your eyes, please. Switch off the UV, switch on the fan or the blowers, switch on the light, and generally you are provided here with a chair or a sitting arrangement where you can sit and do the work. Now, depending on the work you will be performing, there are people who keep different kind of stuff inside the laminar flow hood. So, you have to decide. It is advisable that put minimum stuff inside the laminar flow hood, but again this is up to you and your team to decide what all things you wanted to place. But say for example, this laminar flow hood where we started has to be kept for dissection. If this laminar flow hood for the dissection, so coming back out in the facility, so we decided that we wanted to put a laminar flow hood somewhere in this corner for dissection. Okay. Now, if you want to use it for dissection, there are certain prerequisite what you have to take into account. So, what I will do, I will close in here the today's class, and in the next class from this design, this is how far we have reached. We will resume from here what are the features, what you needed them in that dissection, dissecting laminar flow hood or the laminar flow hood which will be used for dissection, what all things you needed to be provided in and around that facility. Okay. Thank you and thanks for listening to this lecture.